Hello, everyone can hear my voice?
Yes, Ma'am Dewi.
Clear, Ma'am Fani? Hello, Ma'am Gigi. Can we start now? Hello, Ma'am Gigi. Can we start now, Ma'am Gigi? Okay. Hello, can you hear my voice?
Yes, it's clear, Mom Dewi.
Okay, hello everyone. Good morning. Selamat pagi, magandang umaga for all participants here. Let me to introduce myself. My name is Jaya. I'm a member of the Faculty of Technobiology Universitas Atma Jaya, Yogyakarta. And I will lead this online seminar. Thank you for this opportunity. Today is a great day for us. We can join in the third series of the first international webinar collaboration between Universitas Atma Jaya Yogyakarta and Senliu University, especially between the Faculty of Technobiology Universitas Atma Jaya Yogyakarta and Natural Sciences Senliu University of Baguio, Philippines. Thanks God that we still safe and healthy so we can join the great event for today. Good morning all, and thank you for all the attendees and also the panelists and participants. You can join and you can also see this seminar from the live streaming on YouTube, on Teknobio UAJ account and Senliu University account, and also streaming on Facebook of Senliu University. A very special opportunity for me to greet all the attendees here and also special panelists here, especially for the Dean of School of Natural Sciences of Senliu University, Mam N. P. Opina, Magister Medical Technology and Master of Management in Health Service Management. Good morning, Mam N. Okay, and also especially for the Director of Office of External Relations, Media and Communication and Alumni Affairs, Mam Gigi, Mary Grace Keridubai Wong. Hello, Mam Gigi. Okay.
Hello, good morning.
Hello. And special also the Vice Rector for Student Affairs, Alumni and Cooperation of Universitas Atma Jaya Yogyakarta, Mr. Pupung Arifin, Esos MSI. Good morning, Pak Pupung. Okay, and also the great host for today from Faculty of Technobiology, the Dean of Faculty of Technobiology Universitas Atma Jaya Yogyakarta, Dr. Exupansia Mursyanti MSI. Morning, Bu Syanti.
Good morning, Ma'am Dewi.
Okay, and today we will be having a great speakers from Universitas Atma Jaya Yogyakarta and Senliu University. There are three speakers, and the first is Insinyur Pramana Yuda, MSc PhD. He is a lecturer of Faculty of Biotechnology Universitas Atma Jaya Yogyakarta. Good morning, Pak Pram.
Good morning, Bu Dewi. Good morning, everybody.
Yeah, and the second speaker is Kevin Christian Ollard, RNT MSc. He is an alumni of School of Natural Sciences, Senliu University. Good morning, Mr. Kevin.
Hello, good morning, everyone.
Okay, and the last speaker is Kenneth G. Paulo, MSc. He is also an alumni of School of Natural Sciences, Senliu University. Hello, good morning, Mr. Kenneth.
Good morning, ma'am.
Okay, and for the webinar series today, we'll be guided by the two great moderators. The first one is Monica Ruwaimana. She is faculty member of Faculty of Biotechnology, Universitas Atma Jaya, Yogyakarta. Hello, Ma'am Monica. Are you there?
Yes, I'm here. Good morning, everyone.
Okay, and the second moderator is Maida Fatima. Maida Fatima Chan. She is member of School of Natural Sciences of Senliu University. Hello, good morning, Ma'am Maida. Are you there? Okay. Okay, for the beginning, the first international webinar collaboration series today, I think it is good for us here for getting a prayer. So the opening prayer will be led and guided by Mam Lian Pauline Karen. Okay, Mam Lian, now the time is yours. Hello, Mam Lian. Okay.
Kulip di kulilipan, kulip di kulipanan, kulip di kulilipan, kulip di kulipanan. Nailahitan ngama daytoy pabuyak at panagyaman iken ka agyaman kami tiawan patingga nga talaknip ken panagayata mo tunggal maysa uray saan mi nga kaya aglalo daytoy pandemic nga agdama ikan nakamkada iti sirip mo iturong mo kad iti panagtatrabaho ken dagiti adda ti gobyerno tapno makatulong ti padaatao ikatem kadapo iti butbuteng mi ken iti panagdandanagami tasikat mangted ti liwliwami tapno natal na ti panagbiagmi nawaten mi ken Kriston Apo mi kukulip di kulili.
Okay. Thank you very much, Mam Lian, for our some blessing today. And hopefully we get positive power from God. Now we will be having the greeting message from the Dean of Faculty of Biotechnology, Universitas Atma Jaya, Yogyakarta, to Dr. Exupransia Mursyanti, MSc. The term is yours.
Thank you, Mam Dewi. The Honorable Dean of School of Natural Sciences, St. Louis University, Mem N. P. Opina, RMT, MM, HSM, Director of Office of External Relations, Media and Communication, and Alumni Affairs, St. Louis University, Mem Mary Grace Carido Baywang, as well as Director of Partnership and Promotion Office, Universitas Atma Jaya Yogyakarta, Mr. Andre Purwanugraha SAMBA, and the honorable speakers from Faculty of Biotechnology of Universitas Atma Jaya Yogyakarta, Insinyur Pramana Yuda MSc PhD, and speakers from School of Natural Sciences, St. Louis University, Mr. Kevin Christian Olarte, RMT, MSc, and Mr. Kenneth J. Paulo, MSc, and as well as all of participants of the third seminar online. Good morning. I hope that all of us always be blessed with health and strength during this COVID-19 pandemic. In addition, I do hope that this pandemic can be resolved soon. Ladies and gentlemen, welcome back at the first international webinar collaboration series with the title Ethnomedicine and Environmental Health in COVID-19 Pandemic. As we know that this webinar series is the result of a collaboration between Universitas Atma Jaya Yogyakarta, Indonesia, and St. Louis University, Philippines. And so far, we completely accomplished the first international webinar collaboration series. And the selected topic for the third webinar is molecular and biotechnology advances in COVID-19 pandemic. The reason why we choose this topic because the molecular method was very important to reveal COVID-19 and to reveal how to gene sequence was different from previously discovered coronavirus and how to detect it accurately and quickly. And molecular methods are techniques which play very important role for vaccine development. In addition, the Faculty of Biotechnology, Universitas Atma Jaya Yogyakarta, also developed courses related to molecular, such as molecular biology, molecular genetics, DNA technology, etc. Today is the day where hopefully we could achieve what we have planned previously, so that let me now as the Dean of Biotechnology Faculty, Universitas Atma Jaya Yogyakarta, express I deep appreciation and sincerely thanks for the opportunity of collaboration provided by St. Louis University, especially the collaboration with the School of Natural Sciences, St. Louis University. Hopefully, this webinar series could be beneficial for all of us, for both Universitas Atma Jaya Yogyakarta and St. Louis University. I do hope that the collaboration between Universitas Atma Jaya Yogyakarta and St. Louis University could be sustained in the future in the form of either scientific meeting or other form such as discussion forum, research collaboration, college collaboration, or student exchange. For student exchange, it will be followed by our Vice Dean, Mrs. Ines Septi Arsiningtyas, PhD. Finally, I would like to express our gratitude and deep appreciation to all of speakers, participants, and also the committee who contribute in this webinar. Hopefully, this webinar could be fruitful and be done smoothly. Have a nice webinar and thank you.
Okay, thank you, Bu Shanti. And hopefully, as Bu Shanti said, that this seminar will be beneficial for us because now we are learn together, even from different place. And hopefully, this seminar will be done smoothly. Thank you, Bu Shanti. Okay, again, this is the first international webinar series about ethnomedicine and environmental health in COVID-19 pandemic. And today, 19 August 2020, is the third series after the first series on 15 July 2020 and the second series on 7 August 2020. In this series, it means the last series of the first seminar, is about molecular and biotechnological advance in COVID-19 pandemic. Before we are starting for the webinar series, I will be explained some rules of this seminar. Maybe Intan can share the rules on the screen. Okay, this is some rules. That one, all audio and video of participant will be mute during the session. And the second, participants are not available to rename, see, or chat to other participants. The third, participants can ask questions to panelists using Q&A room and will be answered at Q&A session. It will be led by a moderator. To receive a certificate, participants must fill an exit link, an exit ticket. The link will be displayed at the end of the session. Okay, maybe 5 minutes before we are finish this seminar. Okay, I think that is just a short rules in the seminar. And now we will be starting for the webinar series today. We will start for the three speakers, Mr. Pramana, Mr. Kevin, Mr. Kenneth, and we will be led by two moderators, Ma'am Monica and Ma'am Maida. Okay, Ma'am Monica and Ma'am Maida, the term is yours and the screen now is yours.
Okay, good morning, everyone. Magandang umaga. So I'm the first moderator, Monica Ruaimana. I'm a faculty from Universitas Atma Jaya, Yogyakarta. And currently I'm studying doctoral program in University of Oregon in USA. So actually it is evening for me here. So I will moderate this first session for all of us. And firstly, can you change the slide for Pram or Mr. Pram slide? Yeah. So I would like to introduce you to our first speaker, Dr. Pramana Yuda. He is an associate professor from Atma Jaya Yogyakarta University. He's graduated quite a long time ago from Gajah Mada University. He finished his master on Bogor Agricultural University and finished his PhD on tropical ecology and conservation biology in James Cook University in Brisbane, Australia. So he already worked with us in Atma Jaya since 1994 and became our vice dean and our vice rector before. Currently, he is leading environmental biotechnology lab and research group in our faculty of biotechnology. He is actually a quite very prominent and legendary figure in ornithology world in Indonesia. He is a current president of Indonesian Ornithologists Union and a very active member of national and international birdwatcher and citizen science communities. He's also a consultant for some national parks and bird sanctuaries in Indonesia. And as you could guess, most of his research is related to the bird ecology and conservation, especially how to employ molecular biotechnology on this field. So without further ado, I will pass this session to our speaker, Dr. Pram, to give us our presentation titled Molecular Ecology of Endangered Wildlife of Indonesia. So thank you. And Dr. Pram, please take the stage for us.
Thank you, Pumani. Okay, I will share my presentation. Yeah. Okay, good morning, everybody. And thanks again to Monique and also for the organizers that invited me as a speaker on the webinar series and to start the third series of that webinar. Some of you may have already questioning my presentation title. What is molecular ecology? And what is the link between the COVID-19 pandemic with endangered wildlife? So to answer that question, let me start with the outline of my thoughts. First, I will discuss about biodiversity crisis and pandemic race, followed by nature during COVID-19 lockdown, and finally about biotechnology tool for biodiversity conservation. So what is the link between biodiversity crisis and pandemic race? Fossil evidence has suggested that the life on Earth has faced huge losses of species in a relatively short period of time. And of course, this is in geological time scale and known as mass extinction. And the world has experienced 5 mass extinctions before. Many biologists believe that we are now in the 6th extinction era. One of the studies suggests that every year, between 17 and 100,000 species vanish from our planet. They show that this current extinction rates are much more bigger compared to the past extinction rates. One of the predictions said that the current extinction rates are 1 to 10,000 times compared to the past extinction rate. The remnant species are also on high risk to extinction. Global analysis using Red List Index for 4 taxon, coral, birds, mammal, and amphibian reveal the negative trend. You can see from the graph here, all the line is declining. That's suggesting that in the recent years, most species are on risk to extinction. I did analysis in Indonesia scale for birds, also shown the same trend. As one of the biodiversity countries, Indonesia has a very high species diversity. Unfortunately, the level of extinction rate in Indonesia biodiversity is also very high. This table shows the species diversity, endemism level, and the amount of threatened species of the major taxonomic group in Indonesia. And more than 700 animal species and 393 plant species are listed as threatened species in IUCN Red List. Now, so what's the cause of biodiversity loss? There are direct driver and also indirect driver. For the direct driver or direct threats, we can see here that land or sea usings that caused by habitat destruction and then makes the habitat loss and also habitat fragmentation. And then or direct exploitation lead to the overhunting by hunting or fishing, pollution, invasive alien species, and also climate change. And there's also some indirect driver that all links to our value and our behavior on how we link or connect with the environment. This biodiversity crisis also put us as people at risk by encountering new zoonotic disease like we face nowadays, COVID-19. That's all zoonotic disease, that is disease that's transmitted from animal to humans. It truly already exists in animals. Those diseases can evolve to infect humans. Every time we enter pristine wildlife habitat and come into contact with wild animal, either for pet, foods, medicine, or other products, we risk contrasting zoonotic disease. 60% of all infectious disease in human are zoonosis disease. And 70% of them it's emerging infectious disease, like COVID-19. Many researchers believe that the SARS-CoV-2 virus originated from a bat or pangolin that globally most heavily trafficked mammals, or potentially both species. The virus may have spilled over to humans from unknown animals or evolved after infecting people. So how does nature during COVID-19 lockdown? It is good for nature. Many countries around the world went into lockdowns to control the spread of COVID-19. This period has caused unusually reduced human mobility. Global human mobility reduced 90% and have positive impacts on the quality of environment. Study worldwide reveal that environmental pollution is reduced up to 30%. And other study in Southeast Asia scale reveal the similar result. There's also countless posts on social media and also online media over the past few months reporting unusual wildlife encounter, especially from metropolitan area. This suggests that nature has responded to lockdown. There not only seem to be more animals than unusual, but there are also some surprising visitors in the city. As example here, flock of peacocks are spotted in the city of Dubai, and dolphins recently show up in untypical calm waters in the harbor of Trieste, Italy. So is pandemic good for nature? Unfortunately not. There's a misperception that nature is getting a break from human during the COVID-19 pandemic. In fact, many rural areas in the tropics are facing increased pressure from land encroachment or grabbing, lead to the deforestation, illegal mining, and wildlife poaching. People who have lost their employment in cities are returning to their rural homes further increasing the pressure on natural resources, while also increasing the risk of COVID-19 transmission to rural area. There are a lot of reports in increasing deforestation in Asia and also in South America or Latin America. Furthermore, areas that are economically dependent on tourism face reduced resources as tourism has come to halt, resulting the rise in bushmeat or white meat consumption in Africa particularly, but also some part of Asia, including Indonesia. So I hope that short explanation makes clear that the link between the biodiversity crisis and pandemic. To prevent other possible pandemic in the future, we have to change our relation with nature, and we can start with give more support to conservation of biodiversity. At the point, we will discuss about the application of biotechnology on biodiversity conservation. Back to the earlier question about what is the molecular ecology. Molecular ecology is field of evolutionary biology that is concerned with applying molecular population genetic, molecular phylogenetic, and more recently, genomics to traditional ecological questions. And basically, it is the ecological genetic, or it's synonym with ecological genetic. And then if we work with threatened species or endangered species, molecular ecology is related to the field of conservation genetic. The advance of molecular techniques have improved our ability to answer the difficult ecological genetic question, such as unidentified individual or species, identification of parentage, founder of new population, population structure, and even most basic parameter on population size. And the molecular techniques have been applied in Indonesia. Here's some example I will present on the clarification of taxonomy, defining the extent of hybridization, understanding relative level of within and among population differentiation, defining the structure of population and sex ratio or sex identification, and also the potential use in wildlife forensic. Identification of taxa and assessing their evolution relationship is crucial to design of efficient strategies for management and conservation. And failing to recognize the existence of a distinct and threatened taxon can lead to insufficient protection. The first example is about the use of molecular identification on orangutan. Based on morphological systematics, orangutan was considered as one species before, as Pongo pygmaeus, and had two subspecies, Bornean orangutan, Pongo pygmaeus, and Sumatran orangutan, Pongo pygmaeus abeli. Molecular identification using various molecular markers such as allelic variation restriction, nuclear DNA hybridization, protein electrophoresis, and also complete mitochondrial DNA, revealed that orangutan now is considered as two different species. Borneo orangutan, Pongo pygmaeus, and Sumatran orangutan, Pongo abeli. Further study on orangutan Borneo orangutan revealed three subspecies of Pongo pygmaeus. Pongo pygmaeus in the central and western part of Borneo, Pongo pygmaeus Moreo, the north and eastern part of Borneo, and Pongo pygmaeus 1PE in southern part of Borneo. Yeah. And in 2017, an isolated population of orangutan in Sumatra, here around Danau Toba, Toba Lakes, was described as the third orangutan species as Pongo tapanulensis, based on morphological, behavior, and also genomic evidence. So here in the figure, we can see that it's very distinct and different with other two species. And the map shows the distribution of the orangutan. So there's the Sumatran orangutan, Tapanuli orangutan, and here's is Borneo orangutan. The second example is about hybridization on macaca in Sulawesi. You know that in Sulawesi have a lot of macaca. It's about 7 macaque species and distributed all over the island here. Macaca nigra in the northern and Ocreana here, etc. And field observation indicate the occurrence of hybrids between Macaca tonkeana and Macaca marua here in their overlap distribution area zone around that area. So based on molecular analysis using mitochondrial DNA and microsatellite, hybridization between Macaca tonkeana and Macaca marua have been confirmed. One of the tools for population management is population variability analysis. To do so, a lot of data are required, like on this list. But unfortunately, the data is not available or very limited for some endangered species. And it's hard to get fit ecological study on that species. So here I will give some example how the molecular technique can help to provide the data needed for the analysis. Basic information, population size, is not available for some species. So here's example from the elephant, Sumatra elephant. So using non-invasive sampling, using the feces or dung of the elephant, and then combined with capture-recapture modeling, population size of Sumatra elephant was estimated based on 15 microsatellite phenotyping. The same DNA from the dung of the elephant was also used to identify the sex, and then by the assessment the sex ratio of elephant. And the molecular sexing using the chromohelical DNA binding, GH31 gene, have been also applied for other taxon, including birds that we have done here in our lab. It's Java sparrow, straw-headed bulbul, and also maleo. Dung sample or feces sample is also very useful for study on predator-prey interaction. Applying meta-mini barcoding using next generation sequence method. Pak Seno Subrata from UGM revealed that the prey of Java mongoose, one of the worst invasive species in the world, have quite diverse prey, including mammals, birds, amphibians, snakes, fish, and many invertebrates. Unfortunately, there are no biodiversity threats for this species. This is a threat for wildlife. 9% of Sumatra elephant deaths was caused by disease. Molecular identification has been applied to identify disease or parasite in elephants, including tuberculosis, elephant endotheliotropic herpes virus, and trypanosomiasis. I also applied nested PCR to identify the avian malaria in Java sparrow. And we heard that from the news, tiger in the brown zoo test positive for COVID-19. And for sure that the test was used PCR-based test as standards for COVID-19 identification. At population level, analysis of genetic population structure is very important to the management of concerned population. This includes identify population in need for new individual, population that could safely donate, population in need of further demographic or environmental consideration. One study by Andayani using cytochrome and RNA genes show two different clades of silver gibbon in Java. It's interesting here that in the west clade and central clade of Java, that we have to consider the information to manage the population of silver gibbon in Java. We also have studied of some endangered species in Indonesia using the molecular techniques included orangutan, Sulawesi macaque, Komodo, and also for Java sparrow. Illegal wildlife trade along with the habitat loss is the two major threats for that lead to the species to endangered. If you see the picture, in the first picture, some of you may differentiate and can identify directly that this is a hornbill ivory. It's not elephant ivory or deer horn because it's still in the original form of the ivory of the hornbill. But if the ivory is already craft, become the ivory crafting, like that pictures here. So it's very difficult to identify whether this is ivory from elephants, hornbill, or deer horn, or other skulls. So DNA barcoding or other technique can help to identify that ivory. This is as protected species or no. Another problem is that there's a lot of raptor and also parrot birds that been confiscated in Indonesia, particularly in Java as the main market. That species has come from Maluku on other part of eastern part of Indonesia. Now they are in captivity of wildlife rescue center in Indonesia, and waiting to be relocated into the wild. The problem is that we don't know where the bird come from. Ideally, that we have to release or relocate the birds or other animal and their own habitat and the origin of that species. So how can we solve that problem? By doing microsatellite genotyping or other markers such as SNP or single nucleotide polymerization, we can trace the origin by applying assignment analysis. So with my last presentation, and now we in genomic era. So I will finish my presentation by quote this statement, very interesting, that genomics offer novel tool with increased precision and provide completely new possibility. So we hope that by applying genomic, we can have better understanding of our endangered species as a basis for better and more effective conservation. I think the end of my presentation. Welcome to Molecular Jungle. Thank you, Salamat, and terima kasih. And I get back to Monique. Thank you.
Okay, thank you, Dr. Fram, for the awesome presentation on wildlife poaching and how it's causing the COVID-19 pandemic and how we're going to utilize molecular genetic research for wildlife forensic and conservation. So for all the participants, if you have a question, please leave your comment in Q&A. So there's a Q&A box in this Zoom. Please write your comment or question there, or you can also leave it in the YouTube streaming. And also, please don't forget to write your name and also your institution or where you come from, so I can read it. I can say it when I read your question to the panelists. Thank you. And with this, I would like to pass this time to Ma'am Maida Fatima Chan to continue to moderate the next session. Okay, so Ma'am Maida, please take your stage.
Welcome to the first international collaboration between Universitas, Atma, Jaya, Yogyakarta, and St. Louis University for this third and final series entitled Molecular and Biotechnological Advances in COVID-19 Pandemic. So I will be the moderator for the second and the third speakers. So I have worked at St. Louis University, at first as clinical laboratory supervisor, and now as a professor. So I have taken some units for Doctor of Public Health, major in medical microbiology. So let me introduce the first speaker for today. Okay, so the second speaker, should I say. So he was a graduate of Bachelor of Medical Laboratory Science in 2015 in St. Louis University, and also a graduate recently at the University of the Philippines, Diliman, Quezon City, for Master of Science in Molecular Biology and Biotechnology. So he worked as a clinical laboratory assistant in St. Louis University in 2016. He also worked as a senior science research specialist from 2019 up to the present in the Molecular Endocrinology Laboratory in the University of the Philippines, and works as a volunteer training staff and research associate for the UP COVID-19 online learning laboratory course, University of the Philippines. The COVID-19 pandemic continuously takes its toll in our daily lives. So in this lecture, we'll be talking about the principle behind COVID-19 testing and its importance as a key to disease management. Ladies and gentlemen, it is my honor to present the second speaker, Sir Kevin Christian Olarte. Sir Kevin, the floor is yours.
So hello, good morning everyone. Let me share you my screen. Okay, so good morning again. I am Kevin Olarte, and for this part of the webinar series, I'm going to talk about the principle behind COVID-19 testing and its accompanying challenges. So here are are our objectives for this lecture. So first, we will be discussing how to choose the appropriate test and samples for testing, and briefly discuss antigen and antibody testing formats for COVID-19. Also, we will go through the principles and steps of real-time quantitative PCR testing, which is the gold standard for COVID-19 testing. And we will try to interpret some examples we will try to interpret some examples of COVID-19 testing results. And lastly, we will discuss the importance of mass testing. So as we all know by now, COVID-19 pandemic is caused by the novel coronavirus or novel beta coronavirus, formerly known as SARS-CoV-2. This virus is a sense-strand envelope virus, and just like its close relatives, it has a relatively large genome compared to other viruses.
Sir Kevin,
So,
may we see your
yes.
slide, please? Thank you very much.
I'm sorry, I wasn't
Thank you.
able to. Here, can you see it?
Sir, thank you.
Okay, so sorry, here are our objectives again. Yeah. As we all know by now, the COVID-19 pandemic is caused by a novel beta coronavirus, formerly known as SARS-CoV-2. And this virus is a sense-strand envelope virus, and just like its close relatives, it has a relatively large genome compared to other viruses. So the virus is believed to have originated at a wet market in Wuhan, China. And based on sequence analysis, the virus may have been transferred from bats to a still unknown intermediate host and eventually crossed to us humans. In humans, it usually causes a mild. So in humans, it usually causes a mild disease. However, in few cases, it can lead to severe acute respiratory syndrome, similar to that of the 2003 SARS coronavirus. And on March 11 of this year, the WHO formally classified COVID-19 as a pandemic. And now, globally, it has already infected 21 million and resulted to around 770,000 deaths worldwide. In the Philippines, specifically, we currently have around 53,000 active cases, and sadly, we continue to see an increase in the number of new cases daily. So our global and local situation really begs the answer to the question, how can we end this pandemic and really put a stop to the spread of infection? So it's not really trivial, and the only way is through accurate diagnosis and efficient contact tracing. So right now, this is our only key in managing the spread of the disease, since there are still no available drugs or vaccines specific for COVID-19. So we'll be talking about this specific test later, but first, some general consideration to take note of. First and foremost, during the whole testing procedure, we should always strictly follow biosafety guidelines. So generally, for COVID-19 testing, we should adhere to biosafety level 2 practices and rules. This entails the use of correct PPEs, appropriate laboratory setups, and the like. So for complete info on this, you can check the WHO and CDC websites for specific technical guidelines, since some of these get updated from time to time. And last but not the least, I would like to stress on this, the test is only as good as your sample. So poor sample collection and wrong choice of test can lead to erroneous results. And sample collection is very important for diagnostic tests in general. And a lot of test kits have already been developed by different countries for COVID-19. However, this doesn't mean that these tests are 100% accurate all the time. So like in all other diseases, proper and accurate diagnosis always starts with evaluating clinical impressions and sample collection. So here's a typical estimated qualitative representation of the course of disease or COVID-19 infection from possible exposure up until to the recovery to the disease. No? So the solid lines represent the different available methods for PCR and antigen testing using different samples. The most common are the blue and the pink lines representing the use of PCR on respiratory samples. The dotted lines or the dashed lines represent the two types of antibody testing And as we can see, these diagnostic tests, such as PCR and antigen tests, are only optimally effective during the early phases of infection. As you can see here, specifically during the first week of symptoms. So while antibody tests are only valuable during the latter stages of the disease. So these tests are not pretty straightforward in the sense that its effectiveness as a diagnostic tool depends on several factors, including the time of onset of the illness, the concentration of the virus in the specimen, and the quality of the specimen collected from a person. So the key takeaway from this figure is that timing of the sample collection is, as well as choosing the right test, is very important and should be correlated with the patient's history and clinical impressions. So choosing the wrong test or collecting the samples at the wrong window period could lead to false results. So COVID-19 tests are generally divided into two types. We have diagnostic tests like the molecular or antigen test, and which detect the presence of the virus in our samples. And we also have antibody tests, which, as the name suggests, detect the presence of antibodies in our sample. So let's start first with the latter. So in the most basic sense, antibody tests, also known as serology tests, are used to detect the presence of immunoglobulins from blood of patients to know if the patient have already had a past COVID-19 infection. Meaning, these tests should never be used as a replacement for diagnostic tests, since they are not capable of detecting an active infection. No? If we remember the figure a while ago, antibodies are only produced during the latter stages of the infection, towards the recovery phase. So a negative result from this test does not or should be confirmed by using a diagnostic test, ideally by PCR. So that being said, these tests are commonly used in research and policymaking as a surveillance tool to determine the extent of the spread in communities. Another relatively rapid test, which is more valuable in diagnosis, is an antigen test. So this type of test determines the presence of the virus itself by detecting certain viral proteins, such as the spike protein and envelope protein of the SARS coronavirus. So, if you review the general structure of a coronavirus, it is characterized by an envelope with spikes resembling a solar corona. So these are the typical proteins we can test for. So the spike protein, which is the one responsible for recognition and attachment of the virus to host cells. We also have the envelope protein and membrane proteins, which mostly make up and hold the viral envelope altogether. And lastly, we have the nucleocapsid protein, which binds and holds the viral RNA. So these tests are usually performed on nasal and throat swabs and usually take about 1 hour. These are more valuable as a screening test for symptomatic patients. And this is because this is due to the reason that they have a relatively low sensitivity, which makes them unreliable for population testing. So hence the same as with antibody tests, a negative result must be confirmed by PCR testing. So here's the general principle of the two tests we've discussed so far. For antigen test, a synthetic SARS-CoV-2 antibodies usually fix on a substrate. When the virus is present in the sample, these antibodies can capture viral proteins onto the substrate, and upon binding, a second antibody included in the reagent kit can bind to the captured antigens or viral proteins. And this second antibody is usually coupled to a receptor, such as a fluorescent dye or a metallic particle, or an enzyme such as a peroxidase, which is capable of activating a separate reporter molecule. Thus, a resultant reading from the reporter molecules indicates the presence of the virus from the samples. Similarly, in antibody test, instead of the viral proteins, the fixed antibody can capture human immunoglobulins from the sample, and this time, a synthetic protein carrying a reporter molecule can bind to the immunoglobulin and confirm its presence in the sample. So again, this is just a general idea or principle behind these tests. And actually, there's a lot of formats already available for these tests. The ones we discussed are the general principle of lateral flow kits, However, current platforms such as ELISA and ECRIA are also available. These kits need special equipment and are usually automated. And unlike the lateral flow ones, they are much more capable of high throughput testing, meaning they can test a lot more samples in one run. So we now move on to the main test, or the gold standard COVID-19 or SARS-CoV testing. This test is a diagnostic test which targets or identifies the presence of the viral genome itself in the samples. So it is based on in vitro nucleic acid amplification, known as real-time quantitative PCR or RT-qPCR. This is usually performed on nasal and throat swabs. And more recently, kits amenable to sputum or saliva samples have already been developed. So most RT-qPCR kits detect one or more essential genes of the virus, as we will see later. So if you remember, SARS-CoV-2 is an RNA virus, meaning it has an RNA as its genome. So as we know, RNA is highly unstable and susceptible to nucleases due to its single-stranded configuration. So an important note in RT-qPCR testing is to handle samples carefully and practice good laboratory techniques in order to prevent sample deterioration. RT-qPCR has three main stages. The first one is, of course, RNA extraction or isolation. As the name suggests, in this step, we want to completely isolate and purify the viral RNA from cellular debris and other proteins. We do this to increase the sensitivity of the test, since some proteins or other impurities can cause inhibitions and non-specific reactions in the downstream steps of PCR. So extracting a good quality, pure, and high-yield RNA is very important, since again, the test is only as good as the sample. So a low-yield or degraded RNA extract can give us a negative result. There are a few ways to extract and purify RNA from crude samples, but the two most commonly used methods in COVID testing are either based on the spin column or the magnetic beads method. So in the spin column method, viral particles are first lysed using a mix of detergents and proteinases. Upon complete lysis, the lysates are then loaded into a spin column with a silica filter. So upon centrifugation, all cellular debris and viral proteins are spinned down towards the bottom of the tube, while the negatively charged RNA binds to the positively charged silica membrane. After several repeated washes using decreasing gradients of alcohol, the spin column is then transferred into a new tube, and the purified RNA is now eluted out from the silica membrane using nuclease-free water. On the other hand, in the magnetic beads-based method, same as before, viral particles are lysed using detergents and proteinases, and then a short random oligonucleotides coupled to magnetic beads are introduced and mixed in the sample lysates. These oligonucleotides bind to random RNA sequences, and after some time, an electromagnetic force is then applied to the tube containing the lysates to attract the magnetic beads, holding the viral RNA and human RNA. After several washes, the oligonucleotides RNA complexes are then denatured, leaving the purified RNA in the solution. So both of these methods work well if done right. The spin column-based methods are commonly used in the lab since it's cheaper and doesn't require equipment or heavy machinery, unlike the magnetic bead setups, which is the primary method adopted in automation. However, the spin column method is more time-consuming and more hazardous for the personnel due to long exposures to the sample. So now, once the RNA extracted, we can now proceed to the next parts of the test, which is the reverse transcription phase and the actual polymerase chain reaction. So the next two stages are usually done together using a PCR machine. So to understand how the next two stages work, let's review how the conventional PCR or polymerase chain reaction works. No? So the polymerase chain reaction, in its most basic sense, is an in vitro version of DNA replication. So meaning, what we're trying to do is to copy what is normally happening during cellular DNA replication. So with DNA as a template, we use short oligonucleotides called primers to bind specific target genes. And with the use of synthetic DNA polymerase, we try to make millions of copies of that gene in the lab. So basically, RT-qPCR works in this similar fashion However, going back to the procedure, instead of DNA as a starting material, we have RNA. So the next logical step would be to convert the RNA or the extracted RNA into its complementary DNA in a process known as reverse transcription. So this is a process similar to what RNA viruses actually do in hijacking our cells. So here we use synthetic reverse transcriptase enzyme to make complementary DNA copies of the extracted RNA. So with the reverse transcribed complementary DNA, we can now proceed with the RT-qPCR proper, or the real-time polymerase chain reaction. So following the same principles of the conventional PCR, we use specific primers to target specific viral genes to try and detect and quantify those genes in our sample. So target genes such as RDRP, or the RNA-dependent RNA polymerase, the E gene, N gene, and the ORF1AB gene are the usual targets included in test kits. And in addition to the primers for these genes, in RT-qPCR, we use an additional type of oligonucleotide known as a probe, or also known as a Tachman probe. This probe is similar to a primer in the sense that it recognizes specific sequences in the viral DNA or viral RNA genome. But what's special about it is that it is coupled with the fluorophore and a quencher. So this is what makes RT-qPCR different from conventional PCR. So the fluorophore is strategically placed in close proximity to the quencher, making it silent. And this is the reason why we are able to do relative quantifications of the gene copies, as you will see later. So after separating the DNA strands, and the primers and probes bind to their target sequences, this binding then recruits the DNA polymerase to extend and copy the gene sequence of interest. Now, in the process, the DNA polymerase also liberates the fluorophore away from the quencher, causing it to fluoresce. So most kits available include primers and probes to one or more viral genes. So also included in these kits are primers and probes for human genes. So this will act as an internal assay control, which would indicate if the assay really works. So if you remember, during RNA extraction, aside from the viral RNA, we should also be able to extract human RNA from the sample, because after all, the swab is from a human patient. So the inclusion of a primer and probe set for human gene would be able to tell us if the RNA extraction and the RT-qPCR really work, as we will see later. So the amplification of these three genes, two SARS-CoV genes and one internal human gene, happens simultaneously for every sample during the PC reaction. So note, however, that that each gene is targeted by different primers and probes carrying different fluorescent dye. So like in this example, the amplification of the three genes are differentiated by the three dyes, red, green, and blue. So in simple analogy, it follows that every DNA copy amplified and made, for every DNA copy amplified and made, one fluorescent signal can be detected in real time. And after a few cycles of amplification, more and more fluorescent reporters can be detected. This is where the quantification part happens. Based on this analogy, the machine can monitor and quantify in real time how many copies of DNA has been made as the PC reaction progresses. After the PCR run, the machine software will then analyze the amplification curves for each target as a unit of fluorescent signals emitted by the reporter dyes, and assign a value or assign a threshold value based on this curve. So the threshold value or line is a computed value which takes into account the point at which the targets are all in the amplification phase. It is an assigned value which differentiates the background amplification or background noise from the true amplification. Upon assigning this, the machine would now give out measurements or quantities for every target in the form of CT values or cycle threshold values. So basically, the CT values are the points at which the amplification curves crosses the threshold line. As we will see later, the CT value would determine if a particular sample carries the SARS-CoV-2 particles. So a lower CT value, like the green line, would mean an earlier amplification, meaning meaning more starting genetic material, hence a higher viral load. Conversely, a higher CT value like the red line would mean a fewer RNA starting material, thus a lower viral load. So this is a more realistic data that you would get from a qPCR run. So a normal amplification curve is characterized by a sigmoidal curve like this blue line. This S-curve represents the typical stages of PC reaction or polymerase chain reaction. So from the early stages where barely any signal can be detected due to low copies of the DNA, at a particular cycle, the DNA copies reach a point where amplification can be seen and tracked in real time as increasing exponentially until finally the DNA copies reach a saturation point where all the primers and probes and enzymes are used up, thus representing a plateau phase. So the blue line represents a normal amplification curve. The green represents a negative or no amplification curve, meaning the target is not present in the sample. Thus, there is no amplification or exponential phase seen for the green line. The red line represents an erroneous curve. These types of curves happen for a couple of reasons, and this should be disregarded because it doesn't follow the usual sigmoidal curve. So even though it crossed the threshold value and thus will have a CT value, we are not sure if it really is a true amplification or just a result of human or machine error. So for this particular sample, the test should be repeated. Now, knowing the basics of RT-qPCR, let's try to see some examples. So these are the usual kits used here in the Philippines as seen from the table. These are the particular targets for each kit. So for Sansure, it uses probes or primers for the ORF1AB gene, the N gene, and it also uses the RNase-P as the human internal assay control. So for the Gen Amplify, which is the Philippine-made test kit, it uses RDRP and E gene as the target for SARS-CoV-2 and an unspecified human gene as an internal assay control. So let's look at the Sansure. So because it is usually the test kit used by labs here in the Philippines for proficiency testing. So this is a table from the product insert of Sansure. It tells us the specific cutoff values for the controls. It says here that the negative control should have no CT or no greater than or CT greater than 40 cycles. For the positive control, it should have a CT less than or equal to 35 cycles. And this is the table of interpretation that we're going to use to interpret the results later. So the Sansure kit uses 3 dyes, the FAM for the ORF1AB gene, the ROX for the N gene, and Cy5 for the human internal assay control. So for every sample, the machine will read all 3 channels. So first, having no signals for the 3 dyes, especially for the internal control, which is Cy5, means an invalid test. This could be due to a very low yield of RNA from poor extraction procedure since no human RNA was extracted. On the other hand, no amplifications for FAM, ROX, but an amplification for Cy5 means a negative result. A signal for the Cy5 channel indicates that the RNA extraction was successful and the RT-qPCR reaction worked well. The absence of the signal from FAM and ROX simply means that their respective viral RNA targets are not present in the samples, meaning the sample is simply negative for the presence of the viral RNA. So for a sample or patient to be called out positive for SARS-CoV-2, it should have a CT value or amplification for at least either one of the 2 SARS-CoV gene, meaning a sigmoidal curve should be seen for either one of them. And of course, a sigmoidal curve for the human internal assay control gene. So this is a real run using the Sansure kit on an ABI 7500 fast machine. So in order to interpret the results, we should look at the controls first. No? So looking at the criteria set by Sansure, it seems that the controls are working properly. So we have CT values for every channel for the positive control. And these CT values met the criteria, having values less than 35 cycles, as we can see here. And they are shown by one of these curves. In particular, in this particular example, we can't really see which are the amplification curves for the positive control is, but in reality, you can check them one by one using the machine software. So the negative control have undetermined values, which also meets the criteria. So the flat curves for the negative control should be somewhere here below. So with the controls working, let us look at patient sample 1. So we can see that sample 1 has CT values for all 3 dyes, and they're all below 40 cycles, which means that sample 1 for SARS-CoV-2. So to validate these values, we should check if they are true amplifications by looking at their individual curves. So again, we can see it here, but using the software, in reality, we can check for the amplification curves for each dye. So remember that we should always check for the curves and make sure that it follows a sigmoidal curve to make sure that these CT values are correct and acceptable. So some general considerations: always check the compatibility of these kits, of the kits and machines, since some kits are not optimized to be used with some machines. Also, always practice good pipetting technique. The volumes in PCR testing is very essential. It could mean the difference between a true and false result. And also, we should prevent aerosol forming steps such as vortexing, especially during RNA extraction, because the viruses can be carried by aerosols and thus pose additional risks. And lastly, we should always check amplification curves before reporting the results. So here are some common problems encountered in RT-qPCR. Most of these problems may be due to personal errors, like poor pipetting techniques or accidental contamination of reagents. This can lead to erroneous controls. And it is also possible that you forget to add one of the components during the PCR plate preparation, so like the primers or probes, for example. So this would lead to no amplifications in the well. And also, there's the problem with unusual curves like we discussed earlier. And as I have mentioned, if this happens, the test should be repeated. So other tests that are usually used or are currently in development include CT scans or radiographs. Also, we have LAMP or loop-mediated isothermal amplification. This is a method very similar to RT-qPCR. However, it only operates on a single temperature. We also have methods which are currently used in research, such as the use of the CRISPR-Cas13 or Cas12a system. This refers to the adaptive immunity of bacteria to viruses. Initially, they are used as a genome editing tools. However, in recent demonstrations, they have been used in diagnostics as well. We also have mass spec, time of flight for detection of viral proteins, and of course, next generation sequencing, which is usually used to characterize the genetic landscape of the virus in populations. So knowing how COVID testing works, if we go back to the primary question of how can we end this pandemic, the only answer really right now is to test individuals or increase the capacity of testing. Or in short, in other words, mass testing. So if we say mass testing, we don't mean that we should test each and everyone in the population. Instead, when we say mass testing, we call for an increased testing capacity. We need to do this in order to have a timely data that we can use to determine the actual extent of the spread of infection and possibly guide public health decision making. So in order to achieve this in an ideal setting, testing should be free or at least subsidized. It should be accessible, and of course, it should be timely. But in reality, especially here in the Philippines, we are still falling behind due to the lack of capable labs and trained personnel. Also, there's a problem with supply and demand of the test kits globally, and we still lack a systematic and organized way of contact tracing. So in other words, or in order to break these hurdles in increasing testing capacity, here are some proposed measures, such as expediting the accreditation of newly established labs, training more lab personnel, increase manpower. We also have logistical problems with making an organized procurement process and supply monitoring. And we have to decentralize tests and making it more accessible to everyone. So the bottom line really is that right now, the only way for us to put an end or to at least, or to at the very least, slow down this pandemic is to increase the testing capacity, not only here in our country, but also in the global scale. Because to quote Dr. Tedros of WHO, we cannot fight a fire blindfolded. So thank you for listening and have a good day. Thank you.
Thank you very much. Thank you very much, Sir Kevin. So, Ma'am Maida's internet connection is not that good, but we appreciate very much your presentation. And we hope that we will be able to increase really the number of the people who will be tested so that we will be able to conquer this disease. So, Ma'am Maida, are you there? Hi, Ma'am Maida. Okay.
Okay, so for questions, you are supposed to type them in the Q&A tab, and then we will now proceed. Third and final speaker. Let me introduce him to you. So our third and final speaker for today's webinar series was a part-time research assistant data collector of the dengue project of the World Health Organization. So he was a speaker in 2019, Current Trends in Research, a series of seminar workshop for SLU senior high teachers and students, and is currently a part-time faculty member of the School of Natural Sciences in St. Louis. He graduated in St. Louis University, Bachelor of Science in Biology in 2010, and he has an ongoing Master of Science in Molecular Biology and Biotechnology in University of the Philippines, UP Los Baños, to talk about the overview of molecular biology of SARS coronavirus 2, as well as mutations occurring in the SARS-CoV-2 virus, focusing on the spike gene and protein. Without much ado, I welcome you, Mr. Kenneth J. Paulo. Sir Kenneth, please.
Thank you, Ma'am Maida, for that introduction. Ma'am, can you hear my voice?
Yes, sir.
Okay. So I'll just... yeah. So this topic, Ma'am, thank you again for that kind introduction. So this topic that I will be presenting is actually related to the first and second lecture presentation. So I will now be presenting my topic on the molecular biology and some mutational insights based on whole genome sequencing in SARS-CoV-2. So this will be my overview of what I will be talking about. I will have an overview of SARS-CoV-2 and other coronaviruses, the importance of whole genome sequencing, an overview of the genome, and also some of the mutational hotspots or important mutations that we should be aware of. So to start with, these are the group of coronaviruses infecting humans. So 7 human coronaviruses circulating in the human population, which belongs to the alpha and beta coronaviruses. So there are 4 circulating strains of coronaviruses that are shown in blue. So these are the 229A, NL63, 0C43, and HKU1 that were discovered recently after the SARS epidemic. Now, 10 to 15% of the common colds 10 to 15% of the common colds that we experience sometimes are caused by these viruses. Now, the 3 coronaviruses that are boxed in red that have recently emerged into the human population through species jumping or zoonotic transfer, which are the SARS-CoV-1, MERS-CoV, and SARS-CoV-2, which is now the cause of the current pandemic. Now, this will now be the transmission cycle of the coronaviruses. So for SARS and MERS coronaviruses probably came from bats. So the viruses entered the human population not directly from bats, but one or more intermediate or animal hosts are involved. So for the SARS-CoV-1, the intermediate or animal host is thought to be the civet cats. But while for the MERS-CoV, the intermediate animal host are the dromedary camels. So the viruses in bats jumped into these hosts. The viruses underwent rounds of replication in these hosts and acquired mutations, allowing the viruses to easily transmit into the human population. So what about for SARS-CoV-2? There's still a question if what really the intermediate host is. So it is possible that it can be directly from bats, or one or more intermediate hosts are involved before jumping into humans. And another additional information that there are 500 different coronaviruses identified in bats in China. So what does this suggest? It is unlikely that SARS-CoV-2 will be the last coronavirus that will infect the human population, which means that some of these 500 coronaviruses can continue to jump into the human population in the future. So remember that out of these 500 coronaviruses, there were 3 zoonotic transfer of pathogenic coronaviruses that were introduced into the human population within less than 20 years. So these are the possible intermediate hosts of SARS-CoV-2. We have the ferret cats, small carnivores, pigs, as well as the pangolins. So what then are some of the evidence? What are the evidences? Or how did these organisms become possible intermediate hosts of SARS-CoV-2? So researchers have conducted in vitro experiments and have shown that in addition to bats, SARS-CoV-2 can infect cells from small carnivores, like for example, the ferret cats, the wild cats, as well as pigs. Now, what about for pangolins? Now, there are SARS-CoV-2 related viruses were detected in Malayan or Sundan pangolins, or the Manis javanica. So they are illegally imported in southern China. So they are illegally trafficked mammal species, and sad to say that they are already critically endangered. So they are used as a source of food, and as you can see that they have scales that are used in traditional Chinese medicine. So later on, we will see that through molecular systematics that pangolins is one of the candidates for the intermediate host of SARS-CoV-2. So what then are some of the reasons why it is more difficult, or SARS-CoV-2 is more difficult to manage as compared to SARS-CoV-1? Number one is there is still unknown or proven intermediate host for SARS-CoV-2. But for SARS-CoV-1, the intermediate hosts are the civet cats. And it is important that you know the intermediate host because it is important in breaking the chain of the transmission of the virus. Secondly, there is widespread community transmission for SARS-CoV-2. So it's very difficult to control the mobility of humans because this can actually increase now the transmission rate of viruses of the SARS-CoV-2 virus. While on the other hand, SARS-CoV-1, it occurred in a hospital setting. So there is already a physical barrier that will now prevent the transmission of the virus into the outside world. And for SARS-CoV-2, as we all know, there are a lot of abundant or asymptomatic or mild cases. That is why contact tracing is very, very difficult. While on the other hand, SARS-CoV-1, it will only transmit until 24 to 36 hours after symptoms have been developed. So they lack asymptomatic cases, therefore contact tracing is effective. Now, according to Allison et al. In 2009, that during the evolution of a virus, the transmissibility of the virus is usually increases, whereas the pathogenicity become reduced. So like for example, in compared to the transmission rate of SARS-CoV-1, but the pathogenicity of SARS-CoV-2 is lesser as compared to SARS-CoV-1. So there is now inversely proportional relationship here, that if you increase the transmission rate, the pathogenicity is decreased and vice versa. So these are the factors increasing zoonotic transmission. So as you can see, changes in natural environment for agricultural purposes, climate change, habitat degradation, biodiversity loss, wildlife trade, these are brought about by anthropogenic activities, as mentioned in the first lecture for habitat degradation or biodiversity loss. Now for wildlife trades, so for illegal wildlife trade, just in the case of the pangolins or the Malayan Sundan pangolins, that the people that are in direct contact with these wildlife organisms, it is possible that these wild organisms actually carry pathogenic organisms as well as viruses. So these pathogens can be transmitted to humans. That is why we have laws preventing now selling or trading, illegal trading of this wildlife, because aside from preventing for these organisms to become endangered, it is also to prevent now the viruses from being transmitted to the human population. So this is now the COVID cases worldwide. We have, these are the numbers, cases, active cases. And sad to say that in the Philippines, we have the highest number, which is 169,000. And in Indonesia, they have 143,000. So this is as of August 19. So I know that some of you already have an idea on what the SARS-CoV-2 structure, but I will just have an overview again. So the SARS-CoV-2, it is pleomorphic, so it doesn't have any defined structure. So based on cryo-electron tomography, it will now confirm that it is pleomorphic. So it has a helical nucleocapsid that contains now the genome. That is, so the genome is 30 kilobases of positive RNA. So positive sense RNA, which means that it can directly be read by the ribosomes in the cell. Now, the nucleocapsid is important to protect now the genome, because to protect now the genome. So the genome is now encased in an envelope. Now this envelope is taken from the host. So the virus will now steal the host lipids for them to make their own. And with the envelope, there are also spike proteins as represented by yellow structures here. It gives now the coronavirus its name because it looks like a crown. So for in addition to the spike protein, the E protein here, it is important in the morphogenesis or the formation of the viral particle. So the illustration on the right, this is now the picture that will now show the micrograph of SARS-CoV-2. So you see the protrusions or the extensions. These are the spike protein. So the mechanism of entry, once the virus enters the respiratory tract, it will now be in contact with the epithelial cells of our lungs. So the spike protein, as shown in triangle here, is responsible for engaging interaction with the cell receptor. So it looks like a lock and key mechanism wherein the cellular receptor, in this case is ACE2 receptor, is the lock, and the key is the viral glycoprotein. So the ACE2 receptor for SARS-CoV-1 and SARS-CoV-2 are actually the same. So they bind to the same ACE2 receptor. So the binding of the ACE2 receptor of the spike protein to ACE2 receptor is not enough. A second process is needed to successfully allow the virus to enter. So this includes now, or this needs the TMPRSS2, or it is a cellular protease. So it is now involved in the SARS-CoV-2 entry. So this TMPRSS2, or the protease, will now come and cleaves the ACE2 receptor. So this will now activate the fusogenic state of the protein, which means that the envelope of the virus will fuse to the cell membrane of the host and allowing the entry of the virus. So I would like to focus now on the spike protein, because our main focus here actually is the spike protein, even the mutations in the spike protein. So the spike protein is a trimeric protein. So protein with two major subunits. These are the S1 subunit 1 and subunit 2. The S1, the pink part here, as you can see, it contains now the RBD. The RBD is the green one here. RBD, which stands for the receptor binding domain. So that binds now to the ACE2 receptor. This part here, this S1 and RBD is actually variable part, which is common in most viruses because they are under intense evolutionary pressure because of the interaction with the immune system. Now the S2 part here facilitates now membrane fusion. So it's important for the entry process. It is more conserved and it is hydrophobic, so it can interact with the host membrane. So this is the RBD. This is the ACE2 receptor, the gray one on the upper part that is present in the host cell. That's the receptor in the host cell by which the spike protein will bind to. So this shows now, this part here, lower right, this shows amino acid sequence identity of the receptor binding domain of SARS-CoV-2. So as you can see that the amino acid sequence identity of the RBD or receptor binding domain of SARS-CoV-2 is closely related to SARS-CoV-1, wherein the percent identity is more than 50%. So they are 75% homologous as compared to other human coronaviruses, which is less than 50%. Now this shows now polar interaction, upper left, this one, upper right. So this shows the polar interactions between the polar amino acids of the receptor binding domain with the ACE2 receptor. So this is now the ACE2 receptor, the ribbon-like structure here. So these are the amino acids. So the big letters here will now signify or symbolize the amino acid, and the numbers here are the position of the amino acid in the protein. So like for example, for K, that would stand for lysine in the 417th position. Y, that is tyrosine on the 505th position. So we have threonine for this one, letter T, Y again for tyrosine, Q for glutamine, and N for asparagine. So this amino acid will be different from SARS-CoV-1. So there is now a phylogenetic analysis of the receptor binding domain amino acid sequence. So using molecular systematics, it is used to determine now the relationship of SARS-CoV-2 with other viruses isolated from different bat species, and possible intermediate hosts of SARS-CoV-2 was determined based on the RBD amino acid sequence. So as you can see, the RBD, or the receptor binding domain amino acid sequence from SARS-CoV-2 that is now in red, as you can see here, this one here, is closely related to the RBD sequence, RBD amino acid sequence from the SARS-CoV-2 related virus isolated from the pangolins in GD. GD stands for the Guangdong province in China. And when we say GX, that is from Guangxi. So this is one possible evidence that pangolins are the possible intermediate hosts of SARS-CoV-2. Now the other colors here are from viruses isolated from different bat species. Now the study from Lam et al. Also has used now RBD sequence to, and as you can see that SARS-CoV-2 here, the RBD amino acid sequence is still closely related to the RBD sequence from viruses obtained from the pangolins from Guangdong province. Now, other studies have used now other genes to determine what are the possible intermediate hosts. So for Lam et al., they use now the open reading frames 1AB, the spike gene, the envelope gene, the matrix protein genes, and then the N genes. So I would like to focus on the pink part here on the upper part here, and I would like to enlarge that. So as you can see, this is the SARS-CoV-2 related lineage. So based on these different sets of genes. Now the SARS-CoV-2 isolated from patients infected with coronaviruses was isolated and is still closely related with the SARS-CoV-2 related viruses isolated from pangolins, from Manis javanica. So it is possible that pangolins really are the intermediate host of SARS-CoV-2. Now, do we have pangolins here in the Philippines? Yes, we have in Palawan, I think. And we term that as the Philippine pangolins or the Palawan pangolins, also locally known as the balintong. In Indonesia too, they have the Sunda or the Malayan pangolins that are protected in Indonesia since 1931. Actually, you can have your own analysis and you can publish your own data.
Paketnya dari kementerian yang mungkin
your own data. So you can just obtain you can just obtain the sequences from online databases and just have your own software for generating the phylogenetic tree, and then you can have your own analysis here and you can publish your data without performing the wet lab. Now this slide will now show us one of the distinct characteristic of the spike protein is the acquisition of the polybasic cleavage site, or the S1, S2 cleavage site. So this is a part of the spike protein, still a part of the spike protein in between the S1 and S2 domain. This feature is absent in SARS-CoV-1. So this might suggest an increase in viral infectivity or transmissibility. So this cleavage site, the polybasic site, because they are composed of basic amino acids, it is predicted to be cleaved by other cellular proteases, like for example, the TMPRSS2, during the fusion of the virus to the host cell membrane. So what then are one evidence that it can increase pathogenicity, that this S1, S2 cleavage can increase pathogenicity? The insertion in other viruses has been shown to increase transmissibility, just like in pathogenic influenza virus. Now, I know that you've heard that heard the news that SARS-CoV-2 was manipulated in the lab. Now, this sequence is an evidence that it is not, or it was not manipulated in the lab. So as you can see that RMYNO2 here, the color blue just below SARS-CoV-2, virus isolated from bats also have this polybasic sequence. So it is naturally existing. So this will now be the mechanism of entry. I will not focus on this so much, but after successful binding and fusion, the virus will now enter the virus will now enter the host cell and replicate inside and make a lot of copies of the virus. So the virus will undergo replication and the viral particles will be processed using the host machinery. So I will not focus so much in this aspect, but I would like to emphasize on the genome and whole genome sequencing. So when we say whole genome sequencing, it is from the term itself, it is a process wherein the whole genome of this virus is sequenced. So these are the importance of whole genome sequencing in relation to pathogenesis, in relation to this current pandemic. So aside from obtaining now information about the entire genetic information of the genome and how it reveals the complexity and diversity of the genome, it is an important tool in infectious disease epidemiology. It helps identify the source and animal reservoir. It helps understand the transmission history of pandemics and how a pathogen is evolving. And it will also inform us drug and vaccine design to minimize drug and also vaccine evasion. So this will just be an overview of whole genome sequencing. I will not have an exhaustive presentation on that one. So just like the discussion in the second lecture, second part, that you obtain now the samples, you purify now the viral RNA, and after purifying the viral RNA, you're going to input that in the sequencing machine. So that depends. The mechanism of sequencing depends, but depending on the machine. But the main goal here is to sequence now the whole genome. So after sequencing, like for example, next generation sequencing, and that's already one of the newest trend in whole genome sequencing, you will perform, or bioinformatic analysis will now be performed. So in this case, you will annotate the genes. What are the genes present there? You identify the genes. And what are the genes that are pathogenic that will now be interacting with the host, so on and so forth. So after that, they will now upload, or they will now put that in an online database. Now they will now upload or put that in one of the database. In this example, it's the GISAID. So GISAID, it's a database. It's an international website where you can actually check on this one. It's actually, you can check, it's free. It's available publicly. So it's an international website where genomic sequences of the influenza virus and also SARS-CoV-2 are being deposited from different patients around the world. So this way, scientists can now follow the infection around in the future. So if we have enough sequences, we can now correlate clinical parameters. We can now understand if these strains have different clinical characteristics. So like, for example, one strain causes a more severe disease, other is faster to spread through the population. So if you're going to click now the dots here, like for example here, I have clicked it in here to focus now the Philippine isolate. So it will now show you what country was the genome came from. So in here, the Philippines, it came from RITM, or the Research for Intertropical Medicine. And it will also show you the nucleotide mutations here. I'm sorry, it's very, very small, but if you're going to take a look, it will now show you the nucleotide mutations. What are the changes in the nucleotide mutations? Like, for example, for C, it became T, or it became A. Amino acid mutations can also be, you can also look at the amino acid mutations here. So that is for GISAID. So this will now be an overview of the genome. So we have here, it is 30 kilobase long. Once it is deposited inside, so the translation will follow the gene expression rule that are set by the host cell. So the open reading frame 1AB, so this part here, it will actually occupy majority of the genome of the virus. It encodes for NSP. NSP stands for non-structural proteins. This will now encode the enzymes of the virus. So we also have other genes here, the spike gene, the envelope gene M, and also the N. And other parts of the genome, other accessory genes that encodes accessory proteins, like for example, open reading frames 3A, open reading frame 6, so on and so forth. Now, like for example, for the open reading frames 1A, AB. So NSP1, as I mentioned in the second lecture, this is part one part of the genome that is detected through RT-PCR. So NSP1, this part here, suppresses the antiviral host response. So we also have NSP7 that will now make a complex with NSP8 to form now the primase. NSP12, that is the RNA-dependent polymerase that is detected by primers during RT-PCR. And the other, so there are 16 other NSPs. So the other remaining NSPs are involved in the replication of the viral genome. Now, these are now the mutations occurring within the stretch of the genome of SARS-CoV-2. So as you can see that in all parts, there are mutations here. But I would like to focus now on the highest rate of mutation. So the circles here with colors will now signify the frequency rate of mutation. So like, for example, I would like to focus on the highest mutation that is more than 0.10 frequency rate of mutation. So you can see it in the stretch of the open reading frames 1AB and other parts of the gene. But I would like to draw your attention here on the S cleavage site, on the spike protein. I know that you have heard the mutation on the D614G. So using other references, so the spike glycoprotein, the D614G mutation, it has actually the highest frequency rate, 183 among the others, other mutations occurring within the genome of the virus. And this is another illustration. So that's now the D614G. So what is this mutation? So D, this stands for, this is actually an amino acid, that is the aspartic acid. G, which is the glycine. So in the 614th position of the amino acid of the protein, aspartic acid was changed into glycine. So the change here will now alter the SARS-CoV-2 according to references, according to theories, okay? It's not yet proven, okay? It can alter the SARS-CoV-2 behavior. How? So since it is in the spike protein, so it might, okay? It might increase or decrease the transmission rate of SARS-CoV-2. So it can increase the transmission rate. However, we still lack correlation with, or there's still a clinical, we still lack clinical correlation. We still need to relate that clinically to support that it can either increase or decrease now the transmission rate or the infectivity. Now the D614G or the G variant. So if the amino acid is aspartic acid in the 614th, they term that as the D variant. And if it is G, it mutated to G, that is now the G variant. And the G variant, it is dominant variant of SARS-CoV-2 found. It is actually the dominant variant of SARS-CoV-2 found in swab samples across the world. So that is now in the D614G in the spike protein. And I would like to just mention just one, last one, the NSP6. So the NSP6, it has this frequency rate of mutation. So according to Griffoni et al., it has something to do with antigen antibody. Okay, so NSP6 is located in the open reading frames 1AB, 11,083rd position in the nucleotide in the genome. So like, for example, we already have the vaccine that will correspond to the antigen of this. Like, for example, I just illustrated that to make it simpler. So I've illustrated the blue virus, like, for example, that's the old strain. So once this, we have the vaccine, so the vaccine it is injected into our system, then we will now be producing antibodies that is relative to this antigen, so that if this SARS-CoV-2, this old strain will enter our body, the virus will now be neutralized by our antibodies, by our immune system. However, if there is enough mutation in NSP6, this can alter the antigens. So even though we have the antibodies, even though we were injected with the vaccine, we already have the antigen. I mean, we already have the antibodies for the antigen for SARS-CoV-2 for the old strain. But if it will mutate, it is possible that the mutated strain will have a different antigen. So when it enters, it is possible that we can still have or can still be sick. Okay, so that is according to Griffoni et al. These are just hypotheses. This still needs to be confirmed. So should we be worried because of these mutations? It's a normal part of the viral life cycle. So the shifts and changes aren't always a big deal. And the mutation hopefully may lead to a weaker virus. So the changes can be unnoticeable. And the mutation, according to references, it occurs twice a month. So what about for the vaccine? The vaccine can still work. Of course, the researchers know this very well, the mutations very well. They're making vaccines that will be available regardless of the variant, SARS-CoV-2 variant. And to summarize, this will be my last slide. These are already the COVID vaccines on the final phase of the clinical trials. So for the vaccine from Russia, it's not yet on the final phase of the clinical trial. I hope the Philippine government will not yet obtain that. But one of the leading candidates among candidate vaccine is the Moderna in the United States. It is the mRNA vaccine, which means that the mRNA encoding for the antigen that is enclosed in a nanoparticle will be introduced to our system. So our cells will now recognize this mRNA and then translate that to proteins. And the proteins will be the antigen. And our body will now recognize that, and we will now be producing the antibodies for the antigen. Okay, so that's it. Thank you. And I would like to express my gratitude to St. Louis University, to the organizers of this webinar, to Universitas Atma, Jaya, Yogyakarta, to Ma'am Dean Opiña, to Ma'am Raquel, and to Ma'am Arlene Revogio. Ma'am Maida.
Thank you very much, Sir Kenneth, for that very comprehensive talk. Also to Sir Kevin. Thank you very much. We now proceed to the Q&A. I turn over to our first moderator, Ma'am. Thank you.
Okay, thank you, Ma'am Maida. So we already have several questions. We have actually 5 questions to Dr. Pram, but due to limited time and we already like half hour behind the schedule, so I'm gonna only forward 3 questions out of 5. So the first one from Angelica Cortez to Dr. Pram. Do you think the emergence of new COVID-19 virus is related to the biodiversity loss or destruction? In what manners are they connected? That's the first question. Could you answer that, please? Thanks.
Okay, thank you to Monique and also to Angelica Cortez for the question. As I explained before, that SARS-CoV-2 virus is originated from a bat or pangolin. Dr. Kevin also gave more detail about that transmission. And the virus may have spilled over to human from unknown or because of the infecting from other people. So habitat destruction may cause wildlife move to other habitat that's close to human, or otherwise that human can have more access to the isolated habitat before, and then that makes that the context between the wildlife and human it's closer, that it makes the possibility to spill over from the wildlife to human it's bigger. And also hunting the wildlife for food may also cause the spillover virus. You know that bats and pangolin and other wildlife become a popular food in some area like China and Indonesia, also in northern Sulawesi. So this could be also the potential sources of the transmission of the virus. So that's why now there's international pressure to China and also other countries that have wet market or market that provides wildlife for food to be banned or to be closed. So I think it is one of the manner how that wildlife connected or the pandemic connected to the wildlife or biodiversity conservation. Thank you.
Okay, thank you for the answer, Mr. Pram. And we're going to move to the second question from Mr. Aprianto from Wasur National Park in Papua Island, Indonesia. So he asked about the use of molecular technology in conservation, and he said that most of national park doesn't have ability or resource to conducting molecular ecology, as this technique is expensive and require like state-of-art lab. So he agrees that this technology is going to help the conservation, but often is not applicable for them that work in the national park in the field. So he asked to Dr. Pram, do you have any suggestion how to make it more applicable and accessible by people like Mr. Aprianto that work in the field? That's the question.
Okay, thank you, Mas Aprianto from Wasur National Park, Papua, far away from Java. This is one of the places that on my list to visit. I understand that the facility in the field is very limited, also on the scale of human resources. So one way that university can offer to the national park is that we can make joint research or collaboration research, that we here also already have collaboration with National Park in Sulawesi to assess the genetic molecular of the maleo, that is an endangered bird species and endemic in Sulawesi. So what we did is that we trained the field staff to collect the DNA samples. We use the egg cell membrane of the maleo. You know that the post hatched eggs it's left in the nest, so we can use the nest as a DNA material. So yes, of course that we have to follow the good protocol to collect the samples. So we give a training for them, and then after that, they send to our labs and we analyze that. So in the same way, I think we can mix that kind of collaboration. So of course, we have to sit together to plan a good research and a good methodology and design sampling to get a better answer for the question that need to be based off for the conservation in the field. Yeah, I think that's one way that university can provide the service for the community, including the national park. Thank you again, Mas Aprianto.
Okay, thank you, Dr. Pram, for the answer. And let's move to the last question for the first speaker from Jesslyn Gita from State University of Jakarta. And she asks, how can students help in conservation of endangered animals using microecology? Because as we know, some of the campus cannot operate in the lab or doesn't have the facility or probably limited by COVID. So is the student role limited only to socialization to the society? That's the question. Please the answer.
Yeah, thank you, Jessica. That almost similar to the previous question. That yes, that's true. That now we are very difficult to work in the lab because our university is locked down. So the student needs special permits to work in the campus, and also me myself also it's difficult to run the research because there is lack of assistant or student that helps the research. So what we can do, the student that except that you are going to the lab, like the fifth staff in national park, know that eDNA that's from the soil or dung or feces or other material in the environment can be used as DNA sources. So we're following the good procedure on collecting the DNA material. It's one of the participants that the community, including student, can do. Yeah. So you remember what the citizen science that we collect the data usually visually by visual observation, but now we can start with the citizen science by welcoming the student and other community to collect the eDNA and then send to the lab, university lab network or other lab network. That I think it's a good plan, because one of the part is very difficult and very expensive is data collection. So yeah, we have to go to the field, need money and of course time. So if there's a network of volunteers that have trained on collecting the DNA, we got a very important and very fixed data. So that's one of that I think can be done by student, but of course we have to prepare students to get a skill required for the collection. Thank you.
Okay, thank you, Dr. Pram, for all the answer. And with that is the end of question session for the first speaker. And I will pass this time to Ms. Maida. Okay, thank you.
Thank you, Monica. Okay, so in the interest of time, for the Q&A for the second speaker, he has already answered all of your queries. So thank you very much for the questions. And please refer to the Q&A tab for the answers. Now, I have two questions for the third speaker, for Sir Kenneth, coming from John Raymond Torres of DIMSU in La Union, Philippines. So for Sir Kenneth, sir, what do you think about the idea of using intermediate host in creating a vaccine? Thank you, sir.
Thank you, Ma'am Maida. Thank you, Sir Torres. sad to say that for protocols in research, before applying that to humans, researchers will actually use lower forms of organisms, intermediate, like for example, intermediate host, to test first the vaccine before testing it to humans. And aside from that, it can help us explain the mechanism on how these organisms can be resistant to these viruses, on how they produce the antibodies. This will help the researchers in producing vaccines that is needed now, during this pandemic. So that's my answer, Ma'am Maida.
Okay, thank you very much, sir. So you have
I hope
another.
I
Yes, sir.
answered that. Okay.
Okay, sir. So you also have another question from Mr. John Vincent Pelayo. So can CRISPR, CAS,
CRISPR-Cas9.
yes, can
I
be
think it
used
was
as
answered.
okay,
Okay.
sir. So that was already answered. And then there's another one regarding the mutation, because it was posted in the Facebook page of the Philippine
Yeah,
Gene
the
Center.
D614G,
So
yes.
I was also interested about that, and gladly you mentioned about it. So somebody asked a question regarding that one, and regarding this new mutated strain, G614, there are news that state that there is some lack of evidence. However, it is said to be more contagious and more infectious. Can you try to confirm, sir, if this is really something to be cautious about?
The question is correct that there are still no concrete evidences, because there are no clinical correlation that will prove that the D614G variant is more contagious than the others. So we still need more research on that, and to correlate that in clinical samples to see whether it can either increase or it can decrease now the transmissibility. So as of now, we still wait for confirmation or hard evidences that will confirm that it can increase. But hopefully, if the mutation will decrease, we hope that that will happen. That the mutation in the mutation in the spike protein in the 614th position will decrease now the transmissibility of the virus. So we hope that that will happen, Ma'am Maida.
Thank you very much, Sir Kenneth and Sir Kevin, for your comprehensive answers. So we now turn over to the next part of the program. Thank you very much for attending this webinar. Thank you. Okay, so we would like to thank the key people for making this third and final series possible. Okay, from the Universitas Atma, Jaya, Yogyakarta, and St. Louis University. Ma'am, we will now have the closing remarks. Okay, for us to have the closing remarks, may we have our Vice President for Academic Affairs of St. Louis University, Dr. Roberto Arguelles. Please, thank you.
Yeah, thank you, Ma'am Maida. I hope that I can be heard. Is it clear? It's also nice to see my brother there from Partnership and Promotion of Atma Jaya, Yogyakarta, Indonesia. Pa Andre? Yeah, first, of course, I'd like to thank everyone for completing the third series, or the third webinar, which focused on the molecular and biotechnological advances in COVID-19 pandemic. To our Philippine participants, good noon, and to our brothers and sisters in Indonesia, salamat siang. I hope everyone is okay. I would also like, in behalf of our rector, Reverend Father Gilbert B. Sales, we would also like to commend and thank and appreciate our speakers for today. I think I have seen Dr. Pramana Yuda. I hope I pronounced it correctly. Okay, and of course from Universitas Atma Jaya, Yogyakarta, and speakers from St. Louis University, our own Sir Kevin Christian Olarte. Thank you very much, and of course Sir Kenneth Paulo. So I think there's nothing much to say except that to our participants and the organizers coming from Universitas Atma Jaya, of course, this is my second university, and I missed being there. I hope I will be there soon. Rest assured that our partnership through, of course, the able leadership of the rector of Atma Jaya, together with Brother Andre, our partnership between the two institutions will continue. This might be the last of the series, but I assure you that this will not be the last of the webinar series between the Faculty of Biology as well as the School of Natural Sciences. Also, thank to our Dean, Dean Ann Opiña, for spearheading our team from St. Louis University. So to our participants, we are looking forward that your learning in this webinar series will yield scholarly education as well as publication. Again, we commend all the efforts of the hardworking staff of UAJY and of course SLU. Mabuhay po kayong lahat and thank you very much. Success.
Thank you very much, sir.
Okay, thank you very much, sir. Okay, I think there is a next closing greeting from the Vice Rector for Student Affairs, Alumni and Cooperation, Mr. Pupung Arifin, SOS-MSE. To Pak Pupung, the time is yours. Hello, Pak Pupung, are you there?
Okay. Hello, sorry. I forget to unmute the mic. Okay. Thank you, Ma'am Dewi, for the time for me. And yeah, this is the last webinars series, or the third webinar series for cooperation between St. Louis University and Yogyakarta University in biotechnological, and today's about molecular and biotechnological advances in COVID-19 pandemic. I think these webinars can address today's issues that happen in this world. So I do hope this research outcome can help to solve COVID-19 pandemic in this world today. And congratulations for all of the organizers that make this webinar series happen and successfully done. Especially Pak Andre and teams from Partnership Office in Yogyakarta University, and Ma'am Gigi and faculty in SLU University in the Philippines. And my high appreciation for faculty from Yogyakarta University and SLU that involving for make this webinar series happen. And thank you for another good cooperation between Atma Jaya and St. Louis University, the Philippines, regarding webinar series that has been done today and two previous webinars. And if today's collaboration in terms of biotechnological science, I do hope we could exploring next collaboration in other fields of study. So both university have many study program. So I do hope we can exploring webinars or research collaboration and so on in another field of studies. But for this first series, I think this is successfully done. Thank you, Pak Andre, Bu Ines, and teams in Atma Jaya that make this happen. And congratulations also for today's speakers, Mr. Sir Pramana Yuda, and Sir Kevin Christian Olarte, and Sir Kenneth G. Paulo that already share for us today. Thank you once again. Thank you for new knowledge, especially for me that my study program is outside of biotechnology. This is new knowledge for me, and I do hope once again this result, this research result or research outcome can help to solve COVID-19 pandemic. And also thank you for Ma'am Monica and Ma'am Maida as today's moderator. I do hope we can meet another collaboration meeting in other terms of collaboration activities that conducted by Atma Jaya and SLU. Thank you once again and congratulations.
Pak Pupong.
Okay, yeah.
Yeah.
Sir Roberto.
Well, yeah, just in behalf of St. Louis University, of course, we would like to continue our collaboration, and I hope just instruct or inform Pa Andre.
Okay, yeah, absolutely.
Pa Andre will tell me, do not worry, it will happen, okay?
Okay, okay.
So we will continue with our collaboration with facultas, ISIP, economy, engineering, and Hukum.
Yeah.
We have, in the past, we have this collaboration, so we hope to continue, and of course, in other fields,
Yes,
particularly
of course.
in research.
Yeah. Okay.
I think we have, both of us have good, have good source of people or experts in the field of biology. I can assure you that our team here can actually help in terms of coming up with our collaborative research. Terima kasih.
Ya, maraming salamat.
Yeah.
Okay, thank you, Mr. Pupung and Mr. Roberto. Now, to all participants who attend this online seminar, don't forget to fill in the exit link for getting the e-certificate. So maybe Intan will be share the link on the screen. We will inform to all participants, don't forget to fill in the exit link on your screen. It will be shared in a chat box on Zoom and YouTube and also in Facebook. So don't forget to fill the link. And we will inform to you, all participants, that the PowerPoint material will be managed and shared by our committee via email. So once again, don't forget to all participants to fill the exit link to get the e-certificate, and after that you will get the e-certificate business day. Okay, don't forget. And this link will be okay, and this exit link will be closed on 23 at 12 AM in Indonesian time. It means in 1 PM in Philippine time. Okay, once again, thank you for all panelists, thank you for all committee, thank you for Mr. Roberto as a Vice President of St. Louis University, and thanks for the Dean of School of Natural Science, St. Louis University, Ma'am Anne. Thank you for the Dean of Faculty Biotechnology, Universitas Atma Jaya Yogyakarta, Ms. Santi, and also thanks to Ma'am Gigi for the collaboration, and also thanks for Mr. Andre from Partnership and Promotion Office for joining the day, seminar today, and also thanks for Mr. Pupung to give a close remark in this seminar, and also thanks for all participants from Philippines, from Indonesia, and from another country. I think it is so much many participants here. And okay, I will remind you again for all participants, don't forget to fill the exit link to get e-certificate, and this link will be closed at 23 August 2020 at 12 AM of Indonesian time. It means 1 PM of Philippine time. And e-certificate will be sent to registered email within 5 business days. Okay. Okay, thank you very much and see you in our next webinar. God bless us. Stay safe, stay healthy. Everyone, terima kasih untuk kita semua. Selamat siang, magandang hapon. Goodbye, everyone. And thank you very much. See you.
Pak Pram, thank you Pak Pupung.
Ya.
Thank you.
Terima kasih sekali Pak Pupung, Pak Alberto, Pak Andre.
Ya.
Thank you.
Mam Gigi.
Ya.
Selamat Mang Gigi, selamat Mang An.
Thank
you. Thank you.
Thank you po. Thank you, moms.
Ma'am An.
Thank you po.
Thank you,
Thank you.
everyone.
Thanks po.
Till next time.
Bye po.
See you next time.
Will we do next time?
No, next
Of course.
time.
No next.
No,
Next
next
time.
We still having a homework, right? Right, Mom Gigi?
Yes, yes.
My,
Okay,
bro-
bye.
my brother suddenly disappeared.
Yes, because he has another meeting with finance.
Oh my goodness. Okay, thank you very much for everybody. Intan, Yosua, you already take a picture for all the participant also, correct?
Yes, sir.
Okay, so it's good. So we also have documentation. Thank you very much.
And thank you very much.
Ma Monica, thank you very much from around the world.
Sama-sama Pak, you're welcome.
Bye. Bye.
Goodbye, everyone.
Magandang hapon. Thank you, Mama Ida.
Thank you, sir, for this opportunity. Bye
Yeah.
po.
Salamat po. Bye po.
Salamat po.
Thank you, Ma'am Ann. Thank you very much.
Thank you. Thank you for the opportunity.
Yes. Thank you, Ma'am Dewi, Ma'am Stephanie.
Yeah, Ma'am Dewi, good job. Thank you.
Thank you, Intan. Thank you, Yosua. Thank
Mom
you.
Dewi with her baby.
Thank you, everyone. Thank you, everyone. Thank
Yeah,
you.
thank you.
Oke, saya live dulu ya. Saya masih
Sami-sami. Jai,
Ya,
Pak Andre.
makasih,
Have a
Bu.
nice day. Stay
Ya,
safe.
ya.